Hello, everyone. Can everyone hear me okay? Looks good. I will share my screen with everyone. Before we get started today, I wanted to give a few notes on the end of the class. So apologies for not getting to this to you sooner, but here it is. So in our syllabus, I mentioned that there would be 5 quizzes. We were not able to go quite as quickly as I had expected, so instead there will be 4 quizzes. Still, the lowest quiz grade will be dropped, just like it says in the syllabus. Another question people asked was whether our quiz 4, the final, would be an in-class quiz or whether it would be a take-home style quiz like quiz 3 was. It will be a take-home style quiz like quiz 3 was. I will post the final quiz on the first day of the final exam period, and it will be due on the last day of the final exam period. As for the homeworks, there will be 2 more research methods homeworks. Hold on, we have a question in the chat. So Jen asks, will the final quiz be cumulative? No, it will not. It will be on the 2 topics we have studied since the last quiz, which will be correlation and prediction and the chi-square test. So it will not be cumulative, but as always, some of the concepts that we learned about, like hypothesis testing, will reappear, but the exact procedures won't be the same because it will be on new topics. So one thing that I have changed my mind about, let me get to there in a second, but the second research methods homework, the 2 research methods homeworks, first will be an assignment on correlation and prediction. This is an assignment I think will be fairly easy for you if you understand the concepts of correlation and prediction. I'll post it right away. And it will be due on next Tuesday evening. It will be due on the Tuesday at 11:59 p.m. So you'll have just less than a week to do it. And the second research methods assignment will be one that if you are familiar enough with JASP to follow along with my tutorials, you'll be able to do very quickly because it's going to be 7 data sets in our datasets tab on Blackboard, which I'll specify which ones you can choose. You can choose among them, whichever you prefer or find most interesting, and use JASP to analyze them, and then answer a few questions about them that'll be sort of general across them, such as for a correlation style data set, what is the equation of the regression line, which we'll get to today, and for the chi-square tests, sort of what is the conclusion from this kind of tests. So there is currently posted on Blackboard an extra credit assignment, which I felt that would be fair to give as a extra credit assignment for the class because it is fairly difficult. And that still is available as an extra credit assignment, which will count as an entire whole assignment. But I'll also be giving another assignment, which will be simply to listen to a lecture that I gave last year to my class because we had a few more classes. I was able to get a little bit further into the book. And this will be on the analysis of variance, basically a test of more than 2 groups where you're trying to look at the differences between all the groups and then zeroing in to see if there's particular differences among the groups under consideration. And that's also going to be fairly easy, provided you have the time and inclination. It too will count as a full assignment, which if you are concerned about your grade on the assignments, will definitely bump that up. So for today, we're going to continue and finish up with the assignment, or the lecture rather, on correlation and prediction. So correlation and prediction, we're rounding the end of it. Last time, we talked about how correlation and prediction are 2 sides of the same coin. In correlation, you're given a bunch of data, and you plot your data as a scatter plot, and then using either just formulas that we talked about conceptually in software that you'll see in action today, figuring out what the relationship between those within the 2 variables you measured for each of your subjects was. With prediction, you have a model in mind. you have in your head what you expect the relationship between two variables is, and the goal is to predict, based on one variable, what the other variable is going to be without knowing it beforehand. Both of these fall under the umbrella of statistics concept called regression, although regression itself as a concept is much more broad, complex, and beyond my pay grade. Let me just go back to a place where we can, where did we end up? All right, let's actually go back to the basics of prediction. So the second topic in the correlation prediction chapter is predictably prediction. And one of the things that we want to do with prediction is we want to use this concept called the correlation coefficient, which we learned how to calculate using correlation methods. We want to use that in order to predict future data based on the data we have already we already had. So we used a data set where in this case we had 6 students giving 2 scores each, one about the number of hours slept last night and their happy mood on the morning you're giving them the test. So you have 6 subjects, each contributes 2 scores. One of the scores is what we typically call X, our independent variable, and the other one we call Y, the dependent variable. And for a study where you're interested in the relationship between the number of hours a person sleeps and their mood score, it's clear that the hours slept is going to be the independent variable, and the happy mood score is going to be the dependent variable, because you would want to measure the happy mood score and understand how hours slept affects the happy mood score, the dependent variable. And given these 6 different students and their 2 data sets each, we created a model of the relationship between these 2 variables using correlation. So there's some in the chat. Kamisha, Amanda, Caroline, it will not near, I'm very mindful of the fact that there's not much time left in the class and there will be proportionally less work for you. So I am mindful of that. All right, so continuing on with prediction. The goal of prediction is to figure out what if we knew another subject's independent variable score, so for instance, their hours slept, and we wanted to know, based on that information and this model we have about the relationship between those 2 variables, what their mood score would be predicted to be if that model was true. And we called this model a prediction model. And I told you that in a lot of cases, we start with data, we use correlation to create this thing called a prediction model, and then we use this to predict new data. And then we continuously update our model based on the new data that comes in. Because always a model you make based on a small sample is not necessarily going to be representative of how the real world is. So when you get new data that varies, that diverges from the predictions from your previous sample, you can always update your prediction model. The prediction model stuff has a bunch of, Kamisha, a lot of people also commented that the last quiz took them quite a long time. That definitely was not, it was my intent to make it quite difficult. This final will not be nearly that much difficult. I would estimate there will be 1/3 as many questions. So, one of the things that we needed to understand was the standardized regression coefficient. Unfortunately, the standardized regression coefficient has exactly the same Greek letter symbol as something we've previously encountered for the type 2 error rate, the uppercase Greek letter beta. For the kind of correlation and prediction problems that we're going to be doing, it's quite easy to understand the relationship between the standardized regression coefficient and the correlation coefficient R, because they're equal to one another. And what you can actually think about the relationship between R and beta is that R is based on raw score units, or a ratio between raw score units, and beta is based on z-score units. And in this case, there won't be any difference between those, but there may be cases when you see in JASP output, for example, that beta and R will be close but not identical. So there's a formula for predicting the z-score of a criterion variable based on what you know about your model, that standardized regression coefficient beta, and the z-score of the predictor variable. So the z-score of the predictor variable, in the case of the sleep and happy mood study, would be the z-score of a student's hours slept, as well as beta, which was the R-score of the correlation model you had before. So what we have here is a simple way of calculating or predicting what the z-score of your criterion variable will be based on what is known about the correlation coefficient and the z-score of the criterion variable. So beta will equal R for the sake of this calculation. And in order to figure out what the z-score of the predictor variable is going to be, or the criterion variable, is it's going to be the product of beta and the z-score of the hours slept, or whatever your independent variable is. So Emma, I see your comment in the chat. I promise it will take fewer than 6 hours. I recognize that the last exam was quite difficult. That was not my intent, and I will be much more fair this time. So I would be happy to talk about this after class. So, but for now, I definitely I'm going to just minimize the chat briefly so that I can focus on giving the lecture, but I will be happy to discuss this afterward. And here's just a little illustration of the same problem, where we're using that R value that we calculated last time in the context of figuring out the correlation coefficient. But this time, we're using what we calculated for a correlation model in order to predict what a new z-score, what a new score for happy mood would be given a score that was not in our previous data. So for this example, let's say you wanted to predict what the happy mood score of a student who slept 4 hours the previous night would be. In the previous data set of 6 students, there was no student who had exactly 4 hours of sleep. So how can you use the model that we created to predict how much sleep, what happy mood score such a student would have? You begin with our x variable, which is 4 hours. And for the sake of this equation, we need to figure out what the z-score of that x score is. And we use that familiar equation for figuring out the z-score, as well as the standard deviation, which you can calculate just based on that you have a sample of 6, and you'll calculate the sample size and use something like a calculator or Excel, preferably, to figure out the standard deviation. And so the z-score for an x-score of 4 is minus 1.84. To put that into sort of the standard normal distribution language, we would say that in a raw score of 4 hours of sleep is 1.84 standard deviations below the mean. And our regression coefficient was 0.85, which equaled the R value we calculated for that correlation model. And if we multiply 0.85 times the z-score of our predictor variable, we get minus 1.56. And all this is telling us now is that the predicted z-score will be 1.56 standard deviations below the mean for happy mood scores in total. But that doesn't actually tell us what the happy mood score would be. So in order to convert this into raw scores, we would have to sort of use the other form of the z-score equation where you're solving for the raw score rather than the z-score. And here it just gives you the rearranged problem to figure that out. But this is actually the z-score equation, just rearranged to solve for the raw score of your predicted y variable. And here's just an overview of everything we just did. And we find that if we solve for the predicted y-score, we get 1.02. So another way of thinking about this is that for a hypothetical person who has 4 hours of sleep the previous night, given the correlation model that we produced from the original 6 students, you would predict, based on this model, that someone who slept 4 hours would have a happy mood score of 1.02. So one thing I wanted you to notice is that this happy mood score is actually not a possible value if you remember back to the original question where I said that the happy mood scores could only be 0, 1, 2, 3, 4. And this is something that often happens. Even when you have a numeric variable that can only take on certain values, in this case integer values and 0, you will sometimes get predictions of impossible values. That doesn't mean that you've done something wrong, it just means that this kind of analysis using correlation and prediction assumes that your variables are numeric and continuous, meaning that they can take on any possible value between any other 2 values. But sometimes you just know that your scale cannot take on certain values. And another way of thinking about this is to look back to that thing we call the regression line, or line of best fit. And if you were to find the intersection of 4 hours of sleep on the line, and then track that back to the y-axis for happy mood scores, you would get the same value of 1.02. So all of this is an incredibly inefficient way to actually predict stuff based on a correlation model. And also, this is just a way that the textbook shows in order to soften you up a little bit and prepare you for what the actual best way to do this is, which is to simply calculate the properties of the regression line and use the properties of the regression line to simply plug stuff in and figure out what the prediction y would be given a certain value of x. And that's exactly where we're going right now. And this is called the linear prediction rule because it uses the mathematics of figuring out what the coordinates of a line would be. And if you remember back to high school math classes, there was a common equation for the properties of any line, which was y equals either mx plus b or a plus b times x or bx plus a. It looked something like this. And it's no coincidence that the equation of the line of best fit or regression line tracks this sort of mathematical description of lines in general. And in the sake of this regression line, all of the factors and all of the variables relate to things that can relate to topics in correlation and prediction more generally. So our predicted y-score, you're often going to see that represented as this symbol, which is y with this little hat on it. That little symbol above the y is called, you just simply call it y hat. And what that means, the hat means, is that it is being predicted from a model. We don't actually know what y is in reality, and the value of y we're getting is a prediction. It's not like a true value. And then there are some other things that should look familiar. There's this thing called a regression coefficient, represented by little b. And then there's this thing called a regression constant, represented by lowercase a. And then x is simply the x score. So in our case, x would be 4. And now, what we're going to find is that The textbook has a complex way of figuring out what A and B are based on a certain group of data. But this issue of finding the value of A and B are massively simplified by software, which can simply do a lot of the complex math much more quickly and much more efficiently than you ever could. A common source of confusion is that B, this regression coefficient in our linear prediction rule equation, is not the same as the correlation coefficient. This value of B is actually the slope of the line of best fit. It is not the value of R. And sometimes the value of R is the slope of that is the same as the slope, or it might look similar. But it's just an easy source of confusion, so don't confuse this regression coefficient with the correlation coefficient. But there is a relationship between R and B. R is sort of in standardized units, z-scores, and B is in the raw score units of whatever the variable is. I think I misstated this before, so I apologize. R is based on standardized units, and B is based on raw score units. And to give an example of this, we can go back to the same data set we've been looking on, where we can consider for different values of hours slept. Based on that, what would the prediction be for the happy mood score of such a person? We already saw an example of this, where if you wanted to know, based on this correlation model, what the value of Y would be, if you had 4 hours of sleep, we found that to be approximately 1, it was like 1.02. But then what if you wanted to know what the happy mood score of someone with 17 hours of sleep to be? If you actually plug that into the equation, I'll show you how to get these values of A is -3 and B is +1. I'll show you how to get those in a second, but just hold on for a moment. But if you have 17 hours of sleep, which is sometimes you get 17 hours of sleep, it's not an impossible number, you would actually predict based on this correlation model that the person would have a happy mood score of 14. And I actually showed that here. If you had 17 hours of sleep, you would predict a happy mood score of 14. And that actually is an impossible value because the happy mood score only goes from 0 to 8. So what do you conclude about this? What does this tell you about the model that you chose to work with? Because you chose to work with a linear model that assumes a linear relationship representing like a line that represents the data and the relationship between hours slept and happy mood scores, you end up with an absurd conclusion for a value of sleep that is technically possible. You expect someone to get 17 hours of sleep to have a happy mood score of 14. Similarly, if you observe someone with 0 hours of sleep, based on this correlation model, you should expect they'll also have an impossible score, but this time -3. So one of the common conclusions here is that some models, like in this case, some correlation models, they really only work within a small range of values, within the most typical range of values. And when you get extreme values, the model, quote unquote, breaks down. And what this actually means is that the true relationship between the two variables is not, in fact, represented by a line. It's not a linear relationship. It's much more complex than that. We know that people who get 17 hours of sleep are not insanely happy the next day. And people who get 0 hours of sleep are not like tragically depressed very, very consistently. The actual relationship between sleep and mood is complicated, way more complicated than just a line. But the line is often a very good approximation of the relationship between two variables, especially between the most typical range of them. So most people get between like 5 and 10 hours of sleep, and their mood is sort of typically between, say, 2 and 7. And the model works very well, that linear model works very well in predicting typical hours of sleep and typical mood scores. But when you get atypical and extreme values in either of your two variables, the model is no longer quite as good. There's actually an example of what actually happens when you get really, really sleep deprived. I won't spend too much time talking about this, but I think it's an interesting diversion. The world record for the longest amount of time spent awake is held by this man, Randy Gardner, who I think in the 1970s spent 11 nights or over 260 hours awake. He was being monitored by doctors at the time and psychologists, and the people who verify world records were there supervising him. And over the course of the experiment, they did all sorts of psychological tests and physiological tests to see what happens to a person as they get increasingly sleep deprived. And to a lot of people's surprise, for the first couple nights, he was pretty cognitively normal. Like if you give him tests of intelligence or attention, it was pretty good for a couple of nights. But then increasingly, as you might expect, he got impairments in his memory, he got mood swings, so he wasn't able to regulate his emotions as well. And he was especially impaired in the ability to pay attention. When you tried to talk to him, he just got bored instantaneously and forgot what was happening. He also experienced some things we might consider psychosis, like delusions and hallucinations. And interestingly enough, when he was recovering from this long period of sleep deprivation, despite losing over like 100 hours of sleep, he only slept an extra 11 hours in the following days. So it's interesting that the brain and body don't need like an equal number of time to recover from sleep deprivation. If somehow the brain is able to expedite and speed up the recovery so that you don't need to get sort of more sleep, you don't need to recover as much sleep as you lost. So to figure out those values that I plugged in the previous slides, where A is -3 and B is 1, the best way to do it is using JASP. And I've given an example of this JASP calculation in the lecture materials from today. Let me bring those up. Just a second, JASP's being a pest. There it is. So what we have here in JASP is the same data that we've looked at so far. I've kind of skipped over the part of the problem where I turn a spreadsheet into a CSV file and import it. For all of the future examples, I will not have you sort of convert things into CSV files. I'll just give you the files that you'll sort of import directly into JASP. So there won't be any kind of extra work for you there. The important thing is being able to look at the JASP output and figure out what we care about. So if you recall, JASP gives you multiple options in this top panel. And to get this data set here, I have used the regression tab, and I've had to use two different options here. First, I used the correlation option from the regression tab, which is the first one under classical regression. And I also used linear regression, and this is simply because correlation alone does not give me all I need. So let's look at them one at a time. So for correlation, we put both of our two numerical variables, hours slept and happy mood, into our variables column. I requested this thing called Pearson's R, which is the correlation coefficient we've been talking about. We talked about the different alternative hypotheses last time for a equation where you're testing the correlation coefficient. And we talked about different examples of one-tail tests, two-tail tests. And here we have the alternative hypothesis for testing the correlation coefficient of our data set here. There's the alternative hypothesis is that, the alternative hypothesis here that we're going with is a two-tailed test, a non-directional hypothesis, where all we are claiming is that the two variables, our slept and happy mood, are correlated, but you're not necessarily saying they're correlated in a negative direction or a positive direction. So that's the equivalent of a two-tailed test. We're saying it could be either, they could be either positively correlated or negatively correlated, but we're not sure which. Or you could specify that you believe that they're going to be positively correlated, which is like a one-tailed upper test, or negatively correlated, which is like a one-tailed lower test. And the options I chose are to display pairwise, which is just, otherwise JASP might get confused in associating which pair associates with which, and also report significance, because we're doing a test of significance. I wanted to actually show the p-value. Also just give us some descriptives like the sample size, and show us a scatter plot. So if we look at our results for this correlation option, it shows us everything we need about the descriptive statistics. We have our sample size, which is 6. We have our correlation coefficient R, which we previously saw to be approximately 0.85, but JASP gives us an extra decimal point and shows it's 0.853. And JASP has also done a test of this correlation coefficient and found that the p-value is 0.03. And when the p-value is 0.03, if we had an alpha value, if we were doing a sort of test where I told you to do a test of this, and I told you the alpha value is 0.05, this would be enough to cause you to reject the null hypothesis because our p-value is lower than the alpha value. However, if the alpha value is 0.01, this would indicate that we should fail to reject the null hypothesis, since the null hypothesis our results would not be extreme enough to justify rejecting it. The scatter plot is very useful. Unfortunately, this correlation option on the regression thing does not produce the regression line. It's just very annoying. The correlation option in the regression button is simply to do a test of a correlation coefficient, not to figure out the regression line. For that, we had to click linear regression from our regression tab, choose the second option. When we did that, it's a little bit more complex here, but follow along with what I did when you sort of solve these yourselves and interpret the output. We have here to need to specify what is the dependent variable and which is the independent variable. In the context of correlation, you don't have to specify it, but often in your mind you have an idea of which is which. So the dependent variable is the happy mood score. The covariate, which, because it's possible to have multiple independent variables, you can put our one independent variable, the hour slept. And at that point, you just follow along with what I did, where you specify these things called what our model is. The only things we really care about are to model the dependent variable. So we simply put hour slept as our model term. We click to include the intercept. That'll just give us the value of A in our regression coefficient problem. I also had us figure out an estimate, model fit, descriptives, and this will just give us some stuff we need to know that might help us. And I told it to tell us, I didn't even tell it to do it any plots. And everything here below, model method specification, plots, that stuff, don't worry about it. That's just too complicated. All I care about from this linear regression output is the values of A and B in a regression equation. So once again, here was our scatter plot, but there is no line of best fit. Let's skip the model summary for now, but just notice that it gives us a null hypothesis and an alternative hypothesis. The null hypothesis assumes that here it shows capital R instead of lowercase r, but it's actually the same as the correlation coefficient when you only have two variables. And notice that the actual data that we have is represented, the correlation coefficient is given by capital R in this diagram. Ignore this ANOVA summary table because it actually is not an ANOVA problem. So do you remember in the context of our, I told you that JASP would let you figure out what the value of A and B are in this regression coefficient equation. A and B are necessary for figuring out this predicted y-score, given a x-score that you already know. So A is -3 and B is 1. And you'll notice that in this coefficients part of the model, the alternative hypothesis here is -3, which is the intercept, and the hours slept unstandardized regression coefficient, B, is 1. So that is where you get -3 and 1 for your regression equation. And to understand like what the intercept is and why it sort of is called the intercept, do you remember back in high school math, you learned that this equation for a line has a term called the y-intercept, the place on the line where it crosses the y-axis. So the crossover of the y-axis and the regression line is where x equals 0. So I actually, in a previous slide, asked you for a person who had 0 hours of sleep, so x equals 0, what would the predicted y-score be? And we found that if we sort of projected the line down and saw where it crossed over with the y-axis, you would have a predicted y-score of -3, aka a y-intercept of -3. So one of the things that you might wonder is, why is it that this model is like a better, this line is the best way to fit this data? Why is it the best description of the data that we have? Why not the blue line where the angle is slightly different and the red line where the angle is also slightly different? How do we know that this regression equation is the best one to describe this data? So this is about to go into a little bit of the theory behind this linear prediction rule. And once again, we're assuming that everything is going to be modeled by a straight line, though in reality, sometimes it's not. So what does it mean for a model like our regression line equation to actually be the best fit for the data we have? So when we talk about fit, what we're actually asking is, what is the line that kind of maximizes the predictive power of the data that we have? So here's an example of a study that I did, or it's a compilation of multiple studies where I had rats who weighed different amounts, and I had blood glucose measures, a measure of how diabetic that rat is. And it certainly looks like as the rat's body mass increases, its blood sugar level also increases. And here's the regression line for that particular study. And you can see that there is a positive correlation. But how do I know that this line is in fact the best line? I mean, there's some like cluster of data points above and cluster of data points below. How should we assume that there should be an equal number of data points above the line of best fit as below the line of best fit. Could that be a way of sort of fitting a regression equation to your data? Well, let's see. So the regression equation is actually based on what the JASP is actually doing to generate the regression equation is something called the least squares principle. And the least squares principle is something that is very, very difficult to explain in words. And I'm not going to ask you really any questions about it, but I think that it's really, really useful to see how it's actually calculated, because it basically uses geometry to answer a question about statistics. So the least squares, aka the ordinary least squares approach, involves taking a scatter plot, and then here the line of best fit is already drawn. And for each of the data points, it draws a line directly up or down from where the data point is to the line of best fit, and uses that as one side of a square. And this should remind you of something we learned earlier, which was minimizing the number of squares for a variance calculation. And basically, the line of best fit is the line that, if you were to draw it, would result in the smallest total area of squares for all of the data points that you have. So here is just different data sets. Here are the lines, here the data points sort of form a little curvy line, and you can see that data points that are far away from the line of best fit have proportionally larger squares, meaning that they're larger deviation from what is predicted given the line of best fit. And the ones that are closer to the line of best fit have tiny squares, or even impossible to see squares because they're so small. Here's an example of a data set where you have multiple points that seem to be part of a line, and many of them are very close to this line of best fit. And then there's one outlier data point that has a larger square associated with it because it is farther away from that line of best fit. And then here's another example. And the idea is that that is how JASP is calculating what the line of best fit is. It's testing lots and lots of lines, and it's figuring out which is the line that results in the least total area of these squares that are formed. And this is fairly similar to something we've found before. And we call this sort of total squared area the sum of squared error term, or SSE. And this is just something that, if we were to actually go fully into the ANOVA stuff, would come back, but because that's not going to be a part of the main class stuff, it will be that one of the optional extra credit assignments. I won't go into it too far. So let's actually continue with the chi-square stuff. So chi-square, that stuff has also been posted on Blackboard. And chi-square is by far the easiest thing that we're ever going to learn in this class, because it is by far the simplest kind of hypothesis test you can ever possibly do. It is even simpler than the z-test of a sample score, I think. Well, actually, it's easier than a z-test of a sample mean. I'll put it like that. So I want to kind of go a bit slowly with this because I want to make sure that everyone kind of understands it. This will be part of your final exam, but I believe that it's going to be the easiest part of the final exam. So what is chi-square? Chi-square is a bit of a deviation from what we've been learning thus far, where thus far we have always been doing kinds of problems where at least one of the variables is numeric. So even when we were doing sort of independent samples t-tests where there were two different groups and the two different groups had different variables that sort of are the basis of saying they're in one group or another, there was always a dependent variable that was numeric. Here, for the first time, we're going to look at an example of a data set where there's only one variable, and that variable has a categorical type of dependent variable. So here we are dealing with the categorical type variables, in particular, nominal variables. So if you recall, some of the types of categorical variables are things like male versus female, what sort of car you drive, a person's favorite breakfast cereal. And we talked all the time about how nominal type variables were just incompatible with a lot of the statistical methods we've learned so far, because almost all of the types of variables we've learned so far required you to calculate measures of central tendency like the mean or the median. And when you have nominal type variables, like let's say that you have a survey of your class and you have the breakdown by male and female, there's no way to calculate the mean of the variable of sex, because you can't take male and female and sort of figure out what the mean of those nominal type variables are. So to introduce us to how you look at a data set that has categorical variables and understand how surprising is the data set that you have given certain expectations, which is the core of the hypothesis testing, we need to introduce a new kind of symbol, or a new kind of, a new term. And this term is called chi. So chi is the Greek letter that is sometimes written like this, like a capital X that sort of goes underneath the line. And it is written CHI, but is pronounced chi, to be rhymed with high. Some people say chi squared, and that apparently is incorrect. And normally, just to make it very, very clear that the chi symbol is distinct from the capital letter X, it'll be written in this sort of curvy style way. So the problem with categorical variables like male and female, make and model of car, is that there is no measure of central tendency that you can use to quickly understand your data and compare one data set to another. So, for example, if you have the data set with 4 scores, male, male, female, and male, what is the mean of this data? It really has no mean. There's no sense in which those categorical data can have a mean. Means must be numeric, and they require you to have numbers as the basis of your variables. Similarly, your scores on different people's favorite cereal, these kind of data are also incompatible with mean or median for that matter. So you need to have a different approach to understanding this data and figuring out how unusual is the data you have compared to the expectations you might have coming in, which is to say, does your data match some null hypothesis? I shouldn't have put null hypothesis in air quotes because it is exactly what a null hypothesis is, as we'll see. But if you recall back to the beginning of the class, even though there was no way of calculating the central tendency of categorical type variables, there was still a way to organize them for the sake of better understanding them. This was something that we learned about very early on, even before we learned about other ways of descriptive statistics, and this was to make a frequency table. So for the data, male, male, female, male, you can make a frequency table where you have the variable of sex, you have the 2 values that appear in your data set, and you report the frequency of each of those values. You had a total of 4 subjects, 3 reported male, 1 reported female. And you can do the same for cereal. There were 3 values, Fruit Loops, Kashi, granola, and you report the frequency of each of those 3 values. So in order to figure out how would you know a certain data set is surprising, we need to first introduce a concept called goodness of fit. So goodness of fit is the chi-square version of what we previously called the null hypothesis, or it's strongly related to it. So the chi-square test begins with a group of categorical nominal type variables. And it asks the question, based on the data that I have, is this data likely if I assume that there should be an equal number of scores, an equal frequency of scores for every value that I possibly have? So to go back to our previous example, if you are sampling 4 different people, and let's say that you have a population that you expect to be equal male and female, but your sample has 3 males and 1 female. If you know that the population is an equal number of males and females, and you assume that you're randomly sampling, your expected frequency differs from your observed frequency. Your expected frequency, given an equal number of male and females, should be that you observe 2 of each if you sample 4 people. But you actually observed something different. However, is this observation of 3 males and 1 female so strange in a population that is equal parts male and female in a sample of 4? I think in this case, most people would probably say that if you have a sample of equal parts men and women, and you sample 4 people at random, it's not that strange to get 3 males and 1 female, just because 4 is such a small sample. But let's say, for example, instead of sampling 4 people from a population that's predicted to have men and women, you instead sampled 4,000. But you sampled a larger number of people, but the observed frequencies followed the same pattern. Let's say you sampled 4,000 people, and you observed 3,000 male respondents and 1,000 female respondents. If you did that test, and you had 3,000 to 1,000, even though the ratio of male and female responses is the same, the fact that your sample size was 4,000 instead of 4 means that it's very unlikely that your expectation that there was an equal number of males and females in the population was true. So this is the essence of the chi-square test and the goodness of fit concept. You have an assumption about the relationship between the variables and the frequency at which you'd expect them. And based on your expectations about the frequency of each of the variables you're looking at, you can compare that expected frequency to the observed frequency and see whether the fit between your expectations and reality versus, say, your expectations and your observations is either good or not good. And another way of saying your expectations are different than your observations is that your observations are surprising given your expectations. And bringing back the concept of surprisingness, that was actually the core of all the hypothesis testing procedures we looked at, where we would find it surprising if we had a z-score of a sample that was 5 standard deviations above the mean of the population if you predicted a null hypothesis was true. But 5 standard deviations above the mean is not necessarily impossible. It's just very, very surprising. And there are some observations that are so surprising that it justifies the conclusion that the null hypothesis probably isn't true. So let's look at some possible examples. So let's say that you're teaching a class of 40 undergraduates and you record the number of students of each class rank. What is your expected frequency of each class rank? You can give this either as a ratio of like the number of freshmen, sophomore, juniors, and seniors, or you could actually tell me the actual frequencies you would observe in a class of 40 undergrads. All right, so if you have 40 students and you expect an equal number of each in your class, you would expect 10 students of freshmen, 10 sophomores, 10 juniors, and 10 seniors. You expect 25% of each of the 4 class ranks to be represented in your class of 40. So you might actually think, well, if you're teaching a class, and let's say it's an upper level class, you wouldn't expect to see an equal number of freshmen as you do seniors. And if you have a different expectation than equal number of each, that's something that you can actually factor in when doing the kind of chi-square test. So if you are doing a measurement of the numbers of the proportion of male and female students in your statistics and psychology class, what would your expectation of the gender breakdown of the class be? This is a particularly interesting example because this class has only 3 male students of about 24 students. And certainly that goes against, that's a very surprising observation if you begin with the assumption that there would be an equal number of male and female students. So we'll return to the idea that our expected frequencies can actually be different than just expecting all of the different values will have an equal frequency of each. Like let's say if you're teaching in a school where you know that 90% of the students are female, it's not surprising if 90% of your class of 40 students is female. But if you're teaching in a school where it's about 50% male and female, having a class with 90% female students is very surprising. So here's an example from my previous class. If you have a class of 34 students, you observe that there are 10 male students, and then there are 24 female students. Let's say that you are expecting an equal breakdown of males and females. The proportion of males in the class will be 50%, and the proportion of females will also be 50%. If you assume an equal representation of male and female students, your expected frequency will simply be the total number divided by the expected frequency of each of your 2 values, male and female. So we got to 17 because 17 is 34 times 50% for male and 34 times 50% for female. I encourage you to go to the attached Excel file to see how I calculated this. But it's pretty straightforward if you imagine 50% representation of 2 different categories. And then you can calculate the difference between your observations and your expectations for each of the categories. So in this case, the observation was that there were 10 male students and 24 female students. And we find that the expected frequency of both males and females was each 17. So to find that the difference in observed minus expected frequencies, we got -7 by taking 10 minus 17 to get -7. And the difference for the females would be 24 minus 17 to give us +7. And to figure out the actual chi-square term that's going to be the statistic that we test a null hypothesis with, this is the sample statistic that we're trying to calculate. It's simply the squared deviation score, which should look quite familiar considering the other kinds of tests where we also got squared deviation scores. Next, we figured out the difference squared that's weighted by the expected frequency. So this is something that is really mainly interesting simply to understand the theory behind the chi-square analysis. But this equation, which is the difference squared that we just calculated, divided by the expected frequency, so 49 divided by 17, we get two values because we had two different category values. And the chi-square is simply the sum of both of those. 2.88 plus 2.88 equals 5.76. And that is chi-squared. You will not have to calculate this by hand. JASP will help you calculate it. So here's another example. And this is a study where they were interested in the representation of male and female serial characters on cereal boxes. So someone looked at many different cereal boxes across history, and they found that like 996 of the observed cereal boxes had male characters depicted, and 390 had female characters depicted. If your expectation is that there should be an equal number of male and female characters on cereal boxes, or another way of putting that is an equal likelihood that a cereal box would have a male character or a female character, your expectation would be that 693 cereal boxes surveyed would have male characters, and 693 cereal boxes surveyed would have female characters. And what we have here is that you can compute the difference score, just like we did in this previous example. And you have a squared differences, and then you had the differences squared divided by the expected frequency. And what I want to highlight here is that the main difference between this example and the example on the previous slide for my previous semester's class was the number, the main difference is the sample size. Sample size is 34 here, whereas for the cereal box example, it's about 1,300. And when we calculated chi-squared for my class, it was about 5.76, which is, I mean, it's a number, 5.76. Not knowing anything else about like how a sample statistic is calculated, that's like a reasonable number. But here, if we were to calculate chi-squared in the same way we did before, by taking the sum of both of these square differences weighted by the expected frequency, we'd get a chi-square value of something like 270, which I'm pretty sure we have not seen any examples of a sample statistic reaching a number like close to like 270. It seems very, very absurd. And it goes to show that for the chi-square test, it is very sensitive to sample size. And basically any deviation from the expected frequency, if you have a large sample size, the chi-square test is going to give you a very, very significant result. But we'll get back to that in a second. Let's actually see what the chi-square distribution actually is. So just like other hypothesis tests, the chi-square test is based on a comparison distribution. And it is both similar and different from the t-test and the t-distribution. One way it is similar to the t-distribution is that there are multiple chi-square distributions that depend on the number of degrees of freedom of the problem. However, unlike any of the distributions, the z-distribution or the t-distribution, the chi-square distribution, it sort of looks a little bit strange. And see, for degrees of freedom of 1, it looks kind of like this downward curving exponential thing. And then as you increase the number of degrees of freedom, it looks like a sort of hump that sort of travels to the right. So the major difference between the chi-square distribution and the t-distribution is that the chi-square distribution, every single value on the distribution is positive. So the chi-square distribution starts at 0 and goes up. There's no such thing as a negative value of chi-squared. So we just learned how to figure out chi-squared, which is our sample statistic. What we haven't learned is how to figure out what the cutoff score is. So the cutoff score, just like before, depends on what kind of data you have coming in, as well as some other things that you stipulate in advance. The first thing is the number of categories your variable has. And this will determine how many degrees of freedom your your comparison distribution, chi-square distribution, will have. So in the case of our male and female example, whether it's my class or the cereal box example, we have 2 categories. So we have male and female for the variable of sex. So to figure out what the degrees of freedom is, we'll get there in a second, but it's going to be the number of categories minus 1. So we will have a chi-square distribution with 1 degree of freedom. And let's say, for the sake of this problem, the alpha value is either 0.05 or 0.01. So first, to know which chi-square distribution will serve as the comparison distribution, we'll choose the degrees of freedom according to how many categories our variable has, minus 1. So since for the class gender example, there are 2 categories, our degrees of freedom term will be 2 minus 1 equals 1. And for this, we're going to be using the Shiny app. So the Shiny app is, I'll bring it up in a second. There it is. So one thing to notice is that the Shiny app and the chi-square distribution does not give you an option on this left-hand panel that was previously always present for distributions that we were using as comparison distributions. Specifically, it doesn't give you the option to choose a one-tailed upper test, one-tailed lower test, or a two-tailed test. And that's because the chi-square distribution is unique in that it has no negative numbers. You can, whereas before you could predict a one-tailed lower test that your mean will be, that your sample mean is going to be below what is predicted by the null hypothesis. Here, there's no way of predicting that because we're not dealing with means any longer. And every single value on the chi-square distribution is positive. So another way of thinking about this is that there is only one kind of statistical test that you can do with a chi-square distribution, or one kind of tail test, and that is a one-tailed upper test. Because there's going to be a cutoff value, and everything above that will be in the region of rejection, and everything below that will be in the region where you would fail to reject the null hypothesis. So once again, here is where you would find the degrees of freedom. The degrees of freedom for us in this example will be 1 because we had a variable with 2 categories, male and female. 2 minus 1 is 1, but you can choose any degrees of freedom depending on how many categories you have. And once again, down here, you can choose the alpha value. For an alpha value, let's say for an alpha value of 0.05, we can look at the plot and see that the chi-square cutoff will be 3.84, which is to say that any sample chi-square score that is above 3.84 would cause us to reject the null hypothesis and conclude that there is in fact some deviation from our expectation that an equal number of males and females are in the class. Once again, I'm getting a bit ahead of myself. So first, let's go to the cereal box example, because it's a very, very extreme illustration of the effect of sample size. So for the cereal box example, our chi-square score was 264.96. And if you recall, this is what we calculated here, or your textbook calculated here. And 264 came from the sum of these squared deviation scores. So the sum of those is 264.96. So you can see that the sample chi-square score is not just sort of significant enough, it is incredibly extreme in the context of this chi-square distribution. Like, all you would need is like 3.4 to reject a null hypothesis, but we actually got a sample score of 264. And this really is a great demonstration of just how sensitive the chi-square score is to large sample sizes. And if we compare this to the class example, where we had a chi-square score of 5.76 with 34 students, 5.76 is actually still significant enough, or still extreme enough, to reject the null hypothesis that there's an equal number or equal proportion of males and females, because our score of 5.76 would be around here in the region of rejection. But you can see that it's way closer to the cutoff than that serial example where the chi-square sample score was like 264. Here's a step-by-step example of how to do the hypothesis test for the chi-square. It's not important to me that you do this by hand. What's important to me is that you are able to sort of understand the concepts. I will not have you do this by hand. So let's actually go to a chi-square example in JASP. And the chi-square examples in JASP are very, very easy. Let's actually open up the chi-square example of our classroom, and we can see what happens when we change the sample sizes. I'm sorry, guys, just a moment. All right, I don't want to rush this, so let's actually move the demo of the chi-square problems to the beginning of our next class on Tuesday. And the rest of that class will be just a review of the topics for the final exam. But I do want to comment on some people's concerns expressed in the chat about how difficult the previous exam was. I am aware that it was a very difficult exam. I will grade it on a curve, and some of the more difficult questions I will just discount entirely. I'd be happy to have individual meetings with anyone who is concerned about their grades in the class. I would definitely encourage everyone who has not submitted assignments to submit them. There will be a small penalty for submitting things late, but you will get virtually all the points. If you are, please make sure that you email things to me, and if I do not reply, I've received what you have sent me, please continue pestering me. I have all of those things, and I will grade them all. Yes, so I just want to assure you all that I'm very mindful that the workload for this last exam was just unreasonable. I recognize that. I apologize for that. And I will do my best to make sure that the end of the class is sort of apportioned fairly, and the kind of studying slash workload to round out this class is not kind of unreasonable and does not sort of pull you very, very far away from the kind of work you need to do for the rest of the class. Oh, Jenny. So there will be a very, very simple correlation assignment, and that will be the last sort of research method. That will be the last sort of homework-like assignment where you answer questions about a set of problems related to a chapter. Then there will be one more extra credit assignment posted where it is simply a matter of watching one lecture and then answering some basic questions on the theory behind the analysis of variance, as well as the extra credit assignment that has already been posted earlier in the semester. And the final quiz will be on the topics of correlation, prediction, and chi-square goodness of fit test. There will be no more MyStatLab assignments. This correlation and prediction homework assignment is the kind of assignment that would have been a MyStatLab assignment, but all of the MyStatLab questions were just very, very preoccupied with having you do lots and lots of calculations. And what I actually care about in the context of correlation and prediction is how well you can interpret sort of what scatter plots are saying and issues with like outliers and such stuff like that.